All right, everyone, welcome back. We are moving on to our last session of the workshop that deals with phenotype data in ViewPathDB. So I have a few slides and I will go over some of the ViewPathDB phenotypic data sets, how we integrate data, and how we may curate data, and then I'll show you if a demo search in FunGDB, and we will dive into the exercises that are prepared for you. So as you may already know, ViewPathDB integrates different types of data, and that includes phenotypes. These phenotypes may be generated as a result of sort of projects that are carried out in individual labs, or they can be community-driven projects encompassing several institutions. And the data that we will investigate in some of the exercises include high-throughput data. For example, today you'll explore data sets in several databases that includes ToxoDB and PlasmoDB. You will also, for example, investigate the fitness data in ToxoDB, where Toxoplasma gondii parasites that are expressing Cas9 and transfected with single guide RNAs were used to infect human cell culture. And so the fitness and the phenotype scores were determined, and the frequency of a single guide RNA library. So this study determined the fitness and the phenotype scores, and the frequency of single guide RNAs library corresponding to the relative abundance of the parasites carrying the targeted mutations were determined as well. This is also described by a screenshot here on the right, and you will also see this in the tutorial, where the red points along the plot are genes known to be essential under certain conditions, while yellow dots are known to be expendable. So on PlasmoDB, we'll take a look at the data set that comes from the piggyback insertion mutagenesis, where you can mine data using scores of the relative growth fitness costs for mutating a gene in the Plasmodium parasite. So ViewPathDB also integrates data from external resources, and we will take a look at several databases, such as data integrated from PHI-base, which is Pathogen Host Interaction Database, and also triptag.org database. And today in particular, you will explore cellular localization data in TriTripDB, where we integrated data from triptag, and this particular project aims to determine the specific localization of trypanosome proteins within the cell. So let's see. The other type of data that we can integrate includes different phenotypes that fall within the internal curation theme. And we can also accept and integrate data from the community. So if you know of any large-scale phenotypic data sets that should be in ViewPathDB, do please let us know and send us email. So when it comes to internal curation, these can be targeted curation projects for selected reference genomes. For example, in the past, we carried out a targeted annotation of genes that are required for capsule annotation in Cryptococcus neoformans. And we also curate phenotypes from publications, especially those that deal with high throughput mutant analysis, such as people looking at the knockout strains and, for example, how they behave in terms of growing in different media and different temperatures. When it comes to internal curation, the phenotypes can be curated with controlled vocabularies and also using ontologies. We can apply control vocabularies for phenotypes that are described with text, and they will be structured in a certain format. For example, we can reformat the data with the help of ontologies, and in view party B, we use the entity plus quality or E plus Q approach, which is also known as post-composition approach. It utilizes different ontologies that are used to represent entity, which is the phenotype that is being characterized, and also integrates quality information, such as how the entity or the phenotype is being affected by the mutation. You can imagine that for this type of analysis, we can utilize different ontologies, right? So it's for different organisms that describe different types of phenotypes. There are a number of the ontologies have been formed. For example, if you're working on trypanosomes or parasites, you may utilize ontology that is normally used to describe parasitic lifestyle. Or if you're working with fungi or ascomycetes in particular, you may want to use the ascomycete phenotype ontology or APO to describe mutant phenotypes. And these type of phenotypes could be either decreased growth or various abnormalities when encountered if you're observing cells under the microscope, or could be absence of conidia in fungi. Now, there's also a reason why we use a post-compositional approach when it comes to characterizing phenotypes and integrating them in a structured format and eventually transforming them into searchable data sets. This type of structure allows us to have more flexibility when it comes to data exploration, but it also gives you more power when it comes to machine learning or interoperability and cross-references ontologies between different databases. Within ViewPathDB, that means that you can mine various ontologies in different databases, and also you can build complex ViewPathDB queries that include different types of phenotypic data. Right, I also wanted to mention that we integrate community phenotypic data from the community. And this could be community curated data such as large projects, for example, Neurospora crassa gene knockout project that aim to characterize over 10,000 mutants, knockout mutants, and specifically look at their ability to grow in different temperatures or in different media and characterize various aspects of this growth. For Neurospora crassa, this work was conducted by several labs where they collected a lot of metadata about morphology and hyphal extension in fungi. And we also offer ways to submit phenotypic data as user comments or using phenotype collection forms that can collect information in bulk. In terms of user comments, here I want to show a few examples. Before I move on to the user comments, I want to show you examples of how user-submitted information can be represented in FungiDB. And for example, in this case, for the Neurospora genome project, the type of the information is available on gene record pages, where you can essentially navigate to the gene of interest and navigate to the phenotype section and explore the descriptions that are available of the fungal growth at different conditions, and also look at the image data that is available for each of the conditions described. Now, I mentioned user comments, and when you're on the gene record page, and I will switch to the live demo just in a second, user comments are a great way to integrate the most up-to-date information and to alert the community about, for example, updated gene name or perhaps new phenotypic evidence that you have gathered for this gene. In this case, we're looking at the user comment screenshot for one of the Fusarium genes, and this comment specifically notifies that the knockout of this gene has an effect on the virulence of Fusarium graminearum, which is a plant pathogen. And so I'll go back to the gene record page for this particular gene. And the way you can navigate the way you can find user comments, first of all, is that they're visible at the top of the record page right here on the left. User comments are also searchable, which means any information that you enter as text can be searched within the site search deployed at the top. In terms of navigating to user comments, if you click on the link here, it will navigate you to the record page section that contains this information. And you can further explore, for example, the user comment content. And you can also add your own user comment if you have any additional information available. Now, for those particular situations where you have more than just a handful of genes to update, we do offer user comment submission in bulk, and that one can be downloaded from How to Submit Data to ViewPathDB section, which is located under About, Submit Data, How to Submit Data to Us. So if you scroll down the page, the bulk user comment form will be available for download via the link at the bottom. So all you need to do is to fill out the form, send it back to us, and we will do the rest of it for you. So all user comments are always available, linked under your name and available to you for edits, or you can delete it if you wish to do so at a later stage. So if you have any interesting information to share, give it a try and submit a comment today about your favorite gene. User comments are also really great way to showcase the recent publications, and they're a great way to improve the resources available for the gene of interest. Now, I also wanted to mention that in addition to user comments, we do offer other ways to submit phenotypes. And the phenotypes can be submitted as texts or also as structured vocabulary. For example, the phenotype collection form for parasites and fungi is available from the same page as I have just shown you, which is How to Submit Data to ViewPathDB. So if you navigate to the bottom, expand the phenotype collection form section, you will notice that there are two forms that are linked at the bottom. So one specifically deals with parasites, and the type of the information that you can provide are shown as examples within the example section. So this phenotype form has fields that are required and also optional. So if you're working on the data set and you expect to have, let's say, maybe 100 or maybe a few hundred, 10 or a few hundred genes available in terms of providing characteristics for their phenotypes, you can provide this information and we can help you to organize this information in such a way that it is formatted properly and as such can be quickly integrated into ViewPathDB. And notice that this form provides a lot more sort of various examples how you can describe the phenotypes, which includes structured vocabulary, examples of various ontologies that you can use, type of the mutant phenotypes that are commonly annotated, and the structured vocabulary expressions that are used to annotate the phenotypic terms, and also examples of other terms that have been previously used in ViewPathDB data sets to help you navigate the information that you may want to use to describe the phenotypes that you are collecting. And again, these forms are available for parasites and also fungi, but essentially they can be adjusted to any of the organisms supported by ViewPathDB. Okay, now we will switch gears just a bit. And what I will do, I will navigate to a phenotype evidence search in PlasmoDB and to show you the type of the data sets and the type of the searches currently available in this database. Note that while we integrate different types of data, obviously the type of the searches available will vary, and that depends on the structure of the integrated data sets. For example, in PlasmaODB, we have several data sets, and notice that for each data set, we also list the type of the search that is available. And the definition of the search is also described at the top of the page. You can hover over individual sections to highlight sort of the definition of the search to help you to understand what type of the search you will be running if you click on this button. So in terms of the AGS, the Association to Genomic Segments, this is a search for genes which are associated to genomic segments by EQTL. In terms of the CP, and I also will show you a CP search in FunGDB, the search returned genes based on phenotype characteristics. For example, for data sets that do not have phenotype qualifier data, the CP search integrates a filter parameter that you can quickly search the desired search for the desired phenotypic characteristics. And there's two similarity searches. The first similarity search will identify genes which have a similar profile for an experiment, while the second one searches for genes by similarity of association. And so finally, there's also a phenotype text search, which returns genes whose phenotypic descriptions contain your text term. For example, you may want to search for, if it's fungi, for Canidia that have reduced phenotype depending on what you're working with. So, right, you will practice how to set up the searches on PlasmaODB in your tutorial, but before we move on, I will demo one search in fungi. Oh, and one more thing that I wanted to mention, as a matter of fact, when you decide to deploy a phenotypic search and click on the button that opens up an interface that will allow you to set up the parameters of your search, if you scroll down a little bit to the bottom, for quite a few data sets, we have useful descriptions in terms of what is the experiment is about and what you should know when considering setting up this search. In addition, for all phenotypic datasets, and for all datasets in ViewPathDB, we also provide links to the dataset record pages. And for example, if you click on the link here, and I'll open up in a separate tab, the dataset record page tells you a little bit more about who created this dataset, where it was published, and provides additional information that may help you to understand the type of data that is featured within this dataset. Right, so let me show you a quick search on FunGDB, and then we'll move on into actually creating searches using the tutorial. So I'm now on the web page for fungi, and the way I navigated to the phenotype evidence search, I simply hover over the searches menu at the top, and I started to type in the filter box what I want to find, and I was looking for phenotype evidence. So if I click on the phenotype evidence, I arrive on the screen, and we have several data sets available that includes data sets from Phibase, which integrates curated information about genes with known annotation or links to the disease or outcomes of the disease in the host. We also have manually curated data for selected reference genomes. And as I have shown before, we have integrated information for the Neurospora Genome Project. So in this particular case, I'm interested in mining the data that came from Phibase. And I'll go ahead and click on the CP button, and notice that a few options have been brought up within the search. So all of these fields have been annotated to a number of organisms which are listed at the top. But in this case, I'm interested in looking for any gene that has annotation for aspergillosis or different types of aspergillosis causing disease or various sort of outcomes of aspergillosis. So one way to do this, you can look, for example, for disease manifestations, or probably the simplest way will be to look at the disease. So notice that aspergillosis is shown at the top, so I'll go ahead and select this as well. But there are various types of aspergillosis, and also the different metadata can be used to describe this condition. So in order to be sort of extensive in my search and cover various descriptions that may be associated with aspergillosis, I will go ahead and start typing aspergillus at the filter at the top. And I just pre-run this search before demo, so this is why the rest of the fields are selected. But the expected behavior is that when you start typing aspergillosis, you suddenly will notice that additional metadata has been brought up to your attention. And so in my case, I want to select any and all possible combinations to find all genes that have been annotated with aspergillosis in general, invasive pulmonary, or pulmonary aspergillosis. And so when I click on Get Answer, my run will search in the background, and once the page refreshes, I should identify a list of genes that fit that criteria. And so as from this point on, obviously, you can look at the individual genes, you can download the genes, you can run a few additional analysis, or you can also add a step and cross-link your search with other phenotypic data sets. And as I have mentioned, because we are using ontologies that work across multiple organisms, you can actually combine these records with other phenotypes in other species supported by FungiDB. And so imagine that you can grow an extensive query, in this case, combining not only phenotypic data, but also RNA-seq data, SNPs, and et cetera, to find something maybe particularly interesting when it comes to looking at the mechanism of the disease progression in terms of the genes in Aspergillus. Okay, so I think this is it, Amar, from me. Are there any questions that I should answer before we move on?
I think they've been answered. There was a question about user comments and what kind of comments can one enter on a gene page. And just as a reminder for everybody that you as a community member, you have a lot of power, as Eve is showing you right now. You have the power here to add information on the gene page. As soon as you enter the information and click on submit, your information appears on the gene page automatically, and it's indexed for our text search. So this is not something that gets curated. It's really policed by the community. And so obviously you want to be, think about what you're going to put in there. You want to put some comments that are useful, but also these comments need not be, a treatise on the gene, right? It could be simply this gene functions as a kinase in gametocytes, see associated paper, and then you can put in the PubMed ID in the PubMed ID section. It'll pull out the paper for you, and you can click on submit, and there it is. Your comment is now captured, and I bet you somebody is going to come to this gene page one day and see your comment and really appreciate it, because it's going to contain information that is useful and pertains to the gene. And in fact, there are many comments. So Eve, if you run the user comment search, you can return all genes in a database that have user comments. And you will notice in FungiDB, there are thousands, and the same thing in many of our other databases. And again, the nice thing about it is that the person who entered the comment is associated with the comment. So you can always email them and say, hey, Catherine, what did you, I noticed you put this comment on this gene. Did you actually try and clone this gene? Were you able to knock it out, and so forth?
Yeah.
And sometimes, yeah, go ahead, Eve.
Just to quickly add also, user comments in general is a fantastic way to capture community expertise in terms of, because as a researcher, you are essentially the best person to address certain questions. And user comments, when entered, they can capture text, obviously, but they can also capture images, files, a lot of information that is gathered during research, whether you're doing PhD thesis or you're a postdoc, will never actually make it to the publication, because nobody tends to publish the multi sort of pages Excel documents that track phenotypes or track other observations that you have made. So sometimes, but at the same time, this is actually quite useful information to have if you're someone who's just starting working with this organism and you would like to know that this particular mutant does not grow on, let's say, these 5 growth media, et cetera, because of certain things, whether it's metabolically important or some other conditions. So yeah, in terms of just like Amar said, the user comments are searchable. So whether you enter a single user comment or you send us a sort of a huge number of user comments for thousands of genes, all of these are useful for people when they're coming to interpret the function of the gene, especially for those genes that are annotated as unspecified. And as such, you really need to do some digging trying to interpret your RNA-seq results or variant calling data.
Yeah, really good points. Also, some of these comments, we always think of a comment or a result, and that's a human nature. I make a mistake all the time. You're always thinking about positive results. If the result is negative, then I don't, this is not reportable. But in fact, simply saying that you tried to knock out this gene multiple times and failed is good information, right? Because this could suggest that this gene is essential and you can't knock it out. And for another graduate student or postdoc or lab to see that comment is quite useful.
Yep. Another good example is updating gene names. So our curation team works currently with a selected number of genomes for which they update gene names and gene products. But we obviously cannot keep up with publications, as there are publications arising every day. But you as a researcher actually have a really good feel in terms of the change, what new things arise within your community, and updating gene names within gene comments will help users to identify the correct genes as well. For example, if gene A has been called, let's say, a kinase, but it has been recently demonstrated that it's not kinase at all, but let's say some other gene, updating the gene name within the user comments will help people to actually identify the correct information and the publications that are linked to support this evidence.
All right, let's see if there are any other questions. Maybe we'll give people a minute or so, or a few seconds at least, to type any questions or unmute themselves if they want.
All right, seems like we should be good to go. Okay,
yeah, sounds good.
Yep, okay, sounds good. So the phenotypic exercises are linked within the schedule on Vue par TB, so you can just simply click on the link to download the PDF associated with this section. And I think we're ready to break out. Thanks very much, Uli, for posting it in the chat as well. So yeah, Rachel or David, we are ready to break out.
All right.